Okay, good day everyone. So this video talk about ribosome. Okay, ribosome. Ribosomes are non-membranous organelles. Okay, that means that these organelles has no membrane bound. Then the ribosomes are the site for protein synthesis. They occur in large number throughout the cytoplasms. So each ribosome actually consists of two subunits. One is called a large subunit, then another one is a small subunit. So when the large and the small subunit, okay, ribosomal subunit, they assemble together, so they form a functional ribosome. So ribosome actually made up of rRNA, okay, we call it ribosomal RNA, and proteins. So they form a ribosome. So there are two basic types of ribosomes. One is a 70S ribosome, which are found in prokaryote, mitochondria, and chloroplast. Then the other one, which are the 80S ribosome, they are found in all eukaryotes. So they made in human cells and other animal cells, plant cells, fungi, protists, they have 80S ribosomes. Then the bacteria and the mitochondria and chloroplast, they have the 70S ribosomes. Yes, the mitochondria and chloroplast have or contains ribosome as well. So what is the S? S is a unit, verbal unit. So this verbal unit is used to measure the rate of sedimentation in a centrifuge. So the greater the S number, the greater the rate of sedimentation. It is slightly different to density, but in other way, we can say that the density, we can analog, okay? We can analogous to use the density, okay, as a verbal unit. But you must remember the verbal is different to density. Okay. Then eukaryotic cells have two populations of ribosome, even though we say we have an 80S ribosome. So, but in eukaryote or eukaryotic cells, we have two types populations, two populations of the ribosome. One is the ribosome that bound to the endoplasmic reticulum. Another one is the ribosome that are freely suspended in the cytosol. Okay, so the one that bound from the to the rough endoplasmic reticulum. So the polypeptide they form normally will be exported or will be transported to the Golgi apparatus. Okay, then the polypeptide that forms is either will be secreted out of the cell or they will pack into the lysosome. In other way, the proteins or the polypeptides that forms in the free ribosomes, here you see is used for the peroxisome. So maybe you have no idea about the peroxisomes. So we can say that it's the proteins and also the enzyme that form for intracellular use. For example, DNAs. Okay. Then we have many DNA polymerase, and they still got many enzymes, okay, ligase that use inside or for internal use of the cell. So these enzymes are synthesized by free ribosome. So the synthesis of proteins carried out more efficiently by a number of ribosomes moving along the same mRNA at the same time rather than just one ribosome. You can see here, so here 1, 2, 3, 4, 5, 6, okay, they attach to only one mRNA. That means at the same time, at least from this diagram, at least here you see, at the same time, they got 6 polypeptides will be formed, will be synthesized. Okay, so the resulting chains of ribosomes are called polyribosome or polysome. So now this phenomenon is known as the polysome or polyribosome. so that more polypeptide can be formed at the same time. Okay, this is by the polysome or polyribosome. All right. So since this video is talk about the ribosome, so we just focus on the ribosome. We are not focusing on the how the polypeptide is synthesized. So you can see the large subunit, small subunit, so they combine becomes the functional ribosomes. Okay, so that's all for this video. Thank you for the listening.